Hello everyone, I'm Jianxu Chen from the Allen Institute of Cell Science. Here, I'm going to talk about the method and tools that we have developed at the Allen Institute for building computational transfer functions on 3D light microscopy images. At the Allen Institute of Cell Science, we want to understand the cell. We believe we should be able to look at the cell and know what it is doing, and what it did, and what it will do. We believe this is possible because of the strong interlink between cell organization and function. Our goal is to create a state space of cell signatures and to understand that space. We take a holistic approach via 3D live cell imaging. To do this, we build the Allen Cell Explorer Toolkit, which includes everything from our stem cell line and methods to new simulation platforms. We want to pave an accessible and robust road from image to information in a quantitative and interpretable way. By solving our own problems in order to move the science forward, we develop application-specific tools. It turns out that most of these tools generalize very well, so we hope to share this with the rest of the world in an easy and accessible way. Today, I will focus on a key component of cell image generator, which we call transfer function. To study cellular dynamics, and more specifically here to study nuclear shape dynamics, we want to segment nuclear shape in 3D and track them over time. I will focus on segmenting the shape in this talk due to the time constraint. With the fast development of deep learning method, this should be easy, right? But when we look much closer, especially from the side view, geez, this is really hard. It is really hard to find an accurate boundary of the nucleus near the top and bottom. Even I can get some segmentation from such images. I can hardly know whether it is accurate or not. Okay, then, why not just imaging cells with higher resolution? It is indeed much easier to extract the nuclear shape, but this can only happen on a much smaller field of view, and as a result, a cell can easily move out of the FOV, and therefore hard to track for a long time. Also, this could result in unhappy cell due to the increased phototoxicity, and therefore we have to take much shorter movies. So, can we perhaps transform the images computationally and get the best from both worlds? Yes, we can. We developed a deep learning model to computationally transform the 20x images to higher resolution for accurate 3D nuclear shape quantification. We call such deep learning model the transfer function. I will come back shortly about more details of the model. The result of applying this transfer function look nice, including from the side view. So now we can extract nuclear shape more accurately. But are the results actually good? And to be clear, how good is actually good? The key to answer these questions is biological-driven application-specific validation. Remember, our goal is to get accurate nuclear shape in 3D over time. To do this, we use spherical harmonics fitting on the segmented nuclei. Actually, how to get segmentation accurately on the predicted image is another important task. We solved the segmentation problem with our Allen cell structure segmenter, but I won't go into details in this talk. Okay, now we know our goal and what analysis step may be affected by the prediction. Then we know how to validate. We can just compare the spherical harmonic coefficients on the predicted and real images. By comparing 5 key spherical harmonic coefficients that dominate the nuclear shape space, we find that the analysis based on our prediction from 20x images will be statistically significantly similar to the analysis we could get from real 100x images. A key message we want to emphasize here is that no matter what deep learning model is used for denoising, deconvolution, generating image of higher resolution, or whatever, a biology-driven application-specific validation is the key to a successful application. Okay, now back to our method. How we did that? There are many existing papers and open source packages for similar purpose. First of all, we are dealing with 3D images and we cannot directly try out those packages for 2D. Then, there are two big directions, unsupervised like cycle GAN or supervised like conditional GAN. A recent study in machine learning found that cyclogram-type models can cause unexpected transformations, so we choose to explore supervised models. Then, existing supervised models for 3D images are based on simulated or semi-simulated data, not completely real data. But we found that not all scenarios can be easily simulated in order to perform well when doing inference on real data. The success may depend on specific structure and specific modality, so we decide to directly use real data for supervised learning. Then, the biggest challenge comes. The images are not aligned. To deal with this issue, we propose two methods. First, we build an effective 3D registration workflow to prepare aligned images for training. Second, when the images are not well aligned, we build an auto-align module inside the neural network to automatically learn alignment. The results shown in previous slides were using 3D registration. The auto-align method could achieve similar performance. This work was done with collaboration with Hongxiao Wang and Danny Chen from University of Notre Dame. We will put all the details in our paper coming very soon. Our method can be applied to other structures such as nucleoli, while the proteins fibrillary and nucleophosphate that show different levels of structural complexity, and the protein H2B and SMC1A that tag different aspects of chromatin, showing different types of texture in the nucleus. The application is not limiting to 20x to 100x. From 100x, we could train another model to further improve the resolution, comparable to area scan fast images in supervised mode. Also, the application is not limiting to fluorescent. We can also do the transfer on bright field images, which makes it possible to combine our transfer function with our label-free technique. Again, biology-driven application-specific validation is the key step before putting any of this into real applications. More details about the validation framework will be available in our bioRxiv paper by the end of this year. The code will be released together with our segmenter tool. We hope to make it easy for everyone to have their own validated transfer function models in their specific applications. Finally, we would like to thank our founder, Paul Allen, for his vision, encouragement, and support. Thank you.